Welcome to the Cutting Edge. This is a new series where we're going to explore groundbreaking scientific ideas that were, just studies published on PubMed or something, and we're just going to go through them. Not in a pop way or how they do it in the news. We're actually just going to go through the studies, the limitations, and what we can see from them. This can go from, pure medical, psychology, novel philosophical ideas to physics. Let's get started. So today we're going to delve into a revolutionary approach to curing type 2 diabetes, T2D, through personalized endoderm stem cell derived islet tissue transplantation. This cutting-edge therapy addresses the core challenges of diabetes management and offers promising outcomes for patients with impaired pancreatic islet function. Background in diabetes. Diabetes, particularly type 2, is characterized by insulin resistance in peripheral tissues and a gradual loss of islet function due to reduction in beta cell mass or dedifferentiation of beta cells. Traditional treatments include lifestyle changes, oral medications, and insulin therapy, but these approaches often fail to provide long-term solutions and can lead to complications such as diabetic nephropathy. Novel therapy. Personalized endoderm stem cell derived islet tissue. The concept. Recent advancements have focused on using human pluripotent stem cells, HPSCs, to generate pancreatic progenitor PP cells or islet tissues. These cells have shown potential in reversing hyperglycemia in diabetic animal models. However, HPSC-derived cells pose risks, such as the formation of teratomas due to residual undifferentiated cells. This has led to the exploration of intermediate stem cell types like non-tumorigenic human endoderm stem cells, ENSCS, which are more suitable for large-scale generation of islet cells. This case study. It's a groundbreaking pilot study. In a landmark pilot study, a 59-year-old man with a 25-year history of T2D and impaired insulin secretion underwent transplantation of islet tissue, e-islets, derived from his own ENSCS. This procedure aimed to restore his islet function and improve glycemic control. Procedure and clinical outcomes. Preparation and transplantation. E-islets were generated from the patient's autologous ENSCS under GMP conditions. These cells met stringent criteria for morphology, purity, viability, and functionality. The patient received 1.2 million IEQs of E-islets via percutaneous transphanetic portal vein transplantation. Monitoring and results. This patient was closely monitored over 116 weeks. The key clinical outcomes included glycemic control. Significant improvement in glycemic control was observed within 2 weeks of post-transplantation. The mean amplitude of glycemic excursion, MAGE, decreased from 5.50 mmol over liter to 1.6 mmol per liter, and the time in target range, TITR, increased from 56.7 to 99%. Insulin independence. The patient gradually reduced and ultimately discontinued exogenous insulin use by week 11. Oral anti-diabetic medications were also tapered off and discontinued by week 56. Safety. Throughout the follow-up period, no tumor formation was detected, and adverse events were minimal and manageable. Of course, I would like to know exactly what those were, but I didn't see anything. As we have seen in, studies done by Purdue or even, the COVID vaccine studies, statements like this are questionable. So I would want to look more into the safety, but of course, over time, more case studies might give us a better view of that. Implications of future directions, advantages and potential. This study demonstrates the feasibility and efficacy of using personalized ENSC-derived islet tissue for treating T2D. This approach circumvents the limitations of donor organ shortages and the risks associated with HPC-derived cells. It offers a promising pathway for achieving long-term glycemic control and insulin independence in diabetic patients. Future research. While the results are promising, further research with larger sample size and trials involving type 1 diabetes patients is needed to confirm the causative role of islets in achieving glycemic targets. Additionally, exploring the pharmacodynamics of stem cell-derived islets and developing universal islets as off-the-shelf products could broaden the applications of this therapy. Conclusion. Personalized endoderm stem cell-derived islet tissue transplantation represents a cutting-edge advancement in diabetes treatment. By addressing the root cause of islet dysfunction and providing a sustainable source of functional islet cells, this therapy holds the potential to transform the management of diabetes and improve the quality of life for millions of patients worldwide. Thank you for joining us on the cutting edge. For further reading, please refer to the study by Wu et al. 2024 in Cell Discovery for detailed insights into this groundbreaking research. Thank you for watching. And in our next one, we're going to be covering if mushrooms actually have a language. Now, this was an interesting study, so I hope that you tune in. Of course, it's a novel one, and it's highly speculative. Let's get into it, though.